Hello, everyone. My name is Komal Jain, and I work as Director of Bioinformatics at the Center for Infection and Immunity at Columbia University Irving Medical Center. Welcome to my talk. I hope you're staying safe and doing well. Today, I'm going to talk about development of capture sequencing assays for enhanced detection and genotyping of tick-borne pathogens and infectious vertebrate viruses. The first part of my talk is about TBD-Cap-Seq. It's an 11-plex diagnostic assay for tick-borne diseases. Recently, we published it in scientific reports, and if you're interested in more information, please check it out. Some of the common illnesses caused by ticks include Lyme disease, which is caused by bacterium Borrelia burgdorferi, and rarely Borrelia mayonnae. Typical symptoms include fever, headache, fatigue, and a characteristic skin rash called erythema migrans. It is the most common vector-borne disease in the United States. It is transmitted to humans through the bite of infected black-legged ticks. Anaplasmosis and ehrlichiosis are also two closely related tick-borne diseases, which are caused by Anaplasma and Ehrlichia respectively, and they are also transmitted to humans by the bite of an infected tick. Babesia is a parasite that causes babesiosis, and it infects red blood cells causing malaria-like symptoms. In addition to transmission via tick, Babesia can be transmitted from mother to unborn child or through a contaminated blood transfusion. Lyme disease diagnostic suffers from many limitations. Unlike Anaplasma and Babesia that have diagnostic molecular assays, the assays for Lyme disease detection suffers from low sensitivity, and therefore serology is the primary method for diagnosing Lyme disease. So the issue with the serology-based assays are that it can lead to false positives because ELISA test for Lyme disease is highly sensitive, and it can lead to false positives from diseases like syphilis, autoimmune disorders such as lupus, tick-borne relapsing fever, and anaplasmosis. And it can also lead to false negatives because an individual may not have developed antibodies at the time of testing. And also, if an individual is immunocompromised, has co-infection or another illness, false negatives are likely. So in order for Lyme disease to be diagnosed, both ELISA and Western blot test must be positive, and both of these tests are indirect methods of diagnosis. They do not detect the actual Lyme disease bacterium. So therefore, we designed this TBD-Cap-Seq method where we can directly make the diagnosis based on nucleic acid as opposed to serology. So this is how the probes were designed. We took the entire nucleotide sequences of every genomic segment of these 11 agents, which resulted in 106 plasmid and chromosomal sequences. To account for high degree of heterogeneity in Borrelia burgdorferi sensostricto species, we included 3 strains representing disparate OSP3 gene types. And the final probe set consisted of 400,000 probe sequences, and it was manufactured by Roche. These are the agents that are included in TBD-Cap-Seq 11-plex assay. There are 3 Borrelia species, there's Babesia, Anaplasma, Ehrlichia, 2 Rickettsia species, and 2 viruses, namely Powassan and Heartland virus. So we conducted a series of experiments to validate these probes, and the first experiment consisted of 3 C3H mice that were infected with N40 strain of Borrelia burgdorferi. From each of these 3 mice, heart, ear, and bladder tissues were obtained, and 2 replete larval ticks fed on these mice were also used in the experiment. This resulted in a total of 11 samples, which were sequenced using unbiased sequencing as well as TBD-Cap-Seq. So the table here shows the results from this experiment. In the first column are the samples. In the second columns are the CT values from RT-PCR. And here we can see the normalized reads from TBD-Cap-seq and the normalized reads from unbiased high-throughput sequencing. We can see that there's a range of CT values. Low CT values means high pathogen burden, and high CT values mean low pathogen burden. And the last column here shows the fold enrichment. What we can see here is that for low CT values as well as high CT values, the recovery of the Borrelia from TBD-Cap-seq is pretty good, and the fold enrichment ranges from 140 to 9,332. And even when the sample is not detectable from qPCR, we are still able to detect it from TBD-Cap-seq, and the performance is much superior to unbiased high-throughput sequencing. The second experiment used 3C57 black 6 mice. One was infected with N40 strain of Borrelia burgdorferi, one with B31 strain, and one was infected with both. From each of these 3 mice, we extracted heart, ear, bladder, and ankle tissues, and 2 replete larval ticks were used. Of these 2 larval ticks, one was a negative control. A total of 14 samples and a negative control were used. The negative control was the salmon sperm sample, and they were sequenced using unbiased sequencing and TBD-Cap-seq. This table here shows the results from this particular experiment. In the first column, we can see the CT values. Again, as before, there's a range of CT values, and the high CT values means low pathogen burden, and these are not detectable from PCR. So what we can see for these samples is the results from both unbiased and TBD-Cap-seq. And we can see clearly that a lot of these samples were not called from unbiased sequencing, but they are detectable from TBD-Cap-seq. And we can see the fold enrichment is pretty high, ranging from 100 to like 52,000, which is a pretty high number. So even with very high CT values such as 35, 34, 33, TBD-Cap-seq did a good job of detecting the pathogen. One thing to keep in mind is that there's an imbalance of reads here, and some of the samples have more reads than the other samples. For example, this low CT sample has 64 million reads, and this tick sample also has 63 million reads. So basically, this caused fewer reads to be assigned to other samples. And if we can play around with sample preparation and library preparation protocols and cause this distribution to be more homogeneous, we can actually correct for this bias in the read distribution. And since the negative samples are correctly identified as negative, positive samples are correctly identified as positive, thus both sensitivity and specificity of this assay is pretty good. These reads here are the ribosomal RNA reads that map to other bacteria. And this schematic here shows the mappings of the reads to the genomes of the B31 strain. And we can see that these 2, mouse 3 bladder and mouse 1 bladder, they were negative from both qPCR and also unbiased sequencing, are both detectable from TBD-Cap-seq, and this particular sample even has a very good depth and breadth of coverage. So the next experiment was to demonstrate the utility of TBD-Cap-seq in ticks and detection of agents other than Borrelia burgdorferi. So in order to do that, we used 18 individual ticks and 2 tick pools. So there were 10 Ixodes scapillaris adult ticks, 5 Ixodes scapillaris nymphs, 3 Dermacentor variabilis ticks, 2 pools of Embryoma americanum, and each of these ticks were infected with 1, 2, or 3 agents. and they were previously screened using qPCR or unbiased high throughput sequencing. So after sequencing, the reads were put through filtration, de novo assembly, and homology search using BLAST. The samples had already gone through more than 3 freeze-thaw cycles prior to TBD-Capseq. So we were not really expecting great results. And despite that, the fold increase ranged from 20-fold to 74-fold for the 5 Ixodes capillaris pathogens, which we will see in the next slide. And we were able to generate complete genome sequences for Powassan virus from 2 tick samples and a 22 kb Borrelia miyamotoi chromosomal contig. We also detected other closely related species that are not detectable from unbiased sequencing. So these are the first 10 samples from this experiment. We don't have the data for unbiased sequencing, but we do have the data from PCR for these samples. And we can see that anything that was called positive from PCR is also called positive from TBD-Capseq. In addition to the positive from the TBD-Capseq, from qPCR, TBD-Capseq can also identify additional pathogens. So it is very good at identifying co-infections and multiple infections with high sensitivity, because the read counts are pretty good, and we can see that the enrichment is pretty high. These are the next 10 samples, and we don't have the unbiased data for these samples, but for this set of samples, we have both qPCR data as well as the unbiased sequencing data. And we can see that TBD-Capseq did really well in terms of detecting the reads for mapping to Borrelia and also other pathogens, as well as we can see here in the parenthesis that the fold enrichment is pretty good. So this experiment also did very well in detecting more than one type of pathogen and pathogens other than Borrelia burgdorferi. The next experiment was to establish limit of detection, and for that we used Borrelia burgdorferi and Vizia microti samples. We quantified the pathogen and serially diluted them in sterile human blood to generate tenfold dilutions. And for both the agents, we were able to get 90% genome recovery when we had greater than 5,000 copies and 25% genome recovery at 1,000 genomic copies. For Babesia microti samples, for 1 to 10 genomic copies, we recovered almost like 1,000 kb of sequence from each of the 4 chromosomes. And for the Borrelia burgdorferi mappings are summarized here. We can see that the qPCR values are listed here, and the read counts, filtered reads, and the reads mapped and the percentage of reads mapped are listed in these rows. We can see that as the qPCR values are increasing, we are still able to detect the Borrelia burgdorferi reads at a qPCR value of 39.84, and even with one smaller dilution, we are able to detect Borrelia correctly. And these for these particular dilutions, these are reads mapped to ribosomal RNA. So these are not these are not the Borrelia reads. But what we can see here is that at very high qPCR values, the CT values, and the samples that are not detectable from qPCR can be detected with TBD-Capseq. So it has really high sensitivity, and it does a good job of detecting pathogens at low, when the pathogen burden is very low. So we continued with validation in clinical specimens, and we in this particular experiment, we demonstrate the performance of TBD-Cap-seq on pathogens present in human blood cells with acute babesiosis. So we took 14 whole blood samples from patients diagnosed with acute babesiosis and 5 samples that were post-treatment follow-up visit. And we found remarkable enrichment of Babesia reads, and 81 to 99% of the reads mapped to Babesia microti. And the assembled complete genome sequence of the 4 chromosomes from each of these samples could be obtained. There were 3 samples with low Babesia loads, and the follow-up samples were negative. And from these samples, we obtained almost like 100 Babesia non-ribosomal RNA reads. So in the next experiment, which is also validation in clinical samples, we took 18 blood samples from patients with erythema migrans or acute babesiosis, and all of these samples were negative by OSCE qPCR, and only 2 of these were correctly identified. So this table here shows, at the bottom here are the samples, the Babesia samples, which are like really strongly positive, and we can see there's a bias of the reads distribution, where most of the reads are assigned to these 2 babesiosis samples, and the other samples ended up getting fewer reads. We can see that Babesia is identified with very high confidence. The number of reads assigned to Babesia is very high, but all of these samples from Lyme disease here are negative. Only 2 samples were correctly identified, but they are not part of this table. They are there's a separate table which is present in the paper. So even with all the negative samples, we were able to accurately identify 2 samples, and we were very successful in Babesia diagnosis with this sample set. So the conclusions from this are that we were able to demonstrate the simultaneous detection and genotyping of tick-borne pathogen disease agents in a wide range of samples. And we can see that TBD-Cap-seq is markedly superior to unbiased sequencing, and it exceeded the sensitivity of qPCR. It can be further improved with modifications to sample preparation and sequencing protocols, and increasing sequencing depth may also increase the sensitivity. And it can offer valuable insights into our understanding of tick-borne diseases by facilitating analysis of previously undiagnosed samples. The next part of my talk is about Vir-Cap-seq Vertebrate. It's a virome capture sequencing assay similar to TBD-Cap-seq. This was actually published in 2015, and it's very well cited by in other articles, and I'm only going to scratch the surface in this for this particular section of my talk, given the time constraint. But for more information, you can check out this article, and there are several other articles that talk about this assay. So this slide here shows the design process and the validation process for this for Vir-Cap-seq Vertebrate assay. So first, the probes were designed, and in order to design the probes, we downloaded all the viral coding sequences from EMBL database. We designed the probes, and from there we designed a unique probe set. Then the probes were validated by mapping against 100 representative genomes to see that the probes are mapping to these genomes ubiquitously, and also they map only to vertebrate viruses, not to plant viruses or insect viruses or bacteriophages. And after in silico probe validation, we did a probe synthesis where the TM was adjusted, homopolymeric sequences were removed and were trimmed, and the probes were synthesized on glass slide using a glass slide method. So after the probes were synthesized, the probes were tested with simulated samples to look for specificity, sensitivity, and to compare the results with RT-PCR, and also to see the performance in multiplexing and to see if probes get saturated or not. And after that, the results were extended to real samples using to see how well it performs when applied in a realistic scenario. So the first step that I mentioned, which is the in silico validation that was performed on 100 genomes. Here is an example showing only 3 genomes. And we can see that when probes are mapped to the genomes of poliovirus or yellow fever virus or parvovirus, we see pretty good coverage, which is ubiquitous. However, we see the difference in the depth of the probes because some sequences are overrepresented in the databases and they result in higher number of probes, and also some parts of the genome are more diverse than the others and they result in higher number of probes. But overall, it's a good representation because we see that the genomes are fully covered in the coding regions. So the first experiment in this particular validation was to compare unbiased with Vircap-seq vertebrate. There were 2 pools here. The first pool consisted of these 3 viruses, and pool 2 consisted of these 4 viruses. The first pool used lung background and the second pool consisted of blood background. Each pool was run with unbiased and Vircap-seq sequencing using Illumina high-seq, and there were 2 replicate runs for each experiment. So the results from this experiment are summarized here. We can see that these are the results using unbiased sequencing, and these are the results with capture sequencing. And we can see that the number of map reads for each of the viruses is significantly greater with capture sequencing than without for all the agents in this particular experiment. And the fold increase is pretty high, and we are seeing up to 8,000 fold increase in the average coverage. So here we can see a schematic showing the enrichment. Basically, this is the unbiased sequencing. This is Vircap-seq vertebrate, and this is the mapping of the probe to the genome. And here we can see that capture sequencing has done a pretty good job of mapping the reads to the reference genome, and whereas the conventional sequencing is pretty sparse and it's not that doing that well. Similarly for this particular enterovirus genome. So this shows the specificity of Vircap-seq. So here we can see the pools where basically these are the reads before a host removal and after host removal. And when you compare the conventional method to Vircap-seq, you can see that Vircap-seq gives you higher number of reads after host subtraction as compared to the regular method. And in the second pool, it's similar that we get more number of reads after host subtraction. And also we can see the percent of host removed is much less with Vircap-seq than with the regular unbiased sequencing, showing that Vircap-seq has higher specificity. Now, in terms of the limit of detection, here we can see that there are 2 experiments in blood and lung, and we can see the copy numbers ranging from like very low of 10 to 5,000. And we can see that with copy numbers as low as 10, we have some detection, and at a copy number of 30, we have pretty good recovery, like almost ranging from 20 to 30%. In blood and in lung, we are also seeing like almost like 5 to 20% of genome recovery at 30 copy number. But even with lower copy numbers, there's still some detection, showing that it's very sensitive. And this particular result shows that it's even more sensitive than RT-PCR. So here we can see the enterovirus in basically using the standard conventional method and also using Vircap-seq. And here we can see that the serum in the Vircap-seq is performing much better than the standard serum. And also the Vircap-seq blood is performing much better than the standard blood. And overall serum is performing better than blood, and similar for HHV1. And these are the detection limits using qPCR, but we can see that with Vircap-seq Vertebrate, at much lower copy numbers, we have significant detection, showing that it's much more sensitive than RT-PCR. And this is the experiment where we used these set of viruses in blood background, and we did a 7-plex lane with 50,000 copies and a 21-plex lane where there were 3 sets of samples which had 500 copies for each of these 7 agents, 50,000 copies, and also set 3 had copies ranging from 3,000 to 10 million. So the idea here is to see if 21-plex gives us similar results as 7-plex, and that increasing the multiplexing affects the results or not. And here are the results, and we can see that with low multiplexing and high multiplexing at 50,000 copies, the results are quite similar. And even with 500 copies, the results are pretty good. Like, we are still able to detect all of these genomes with a good sensitivity. So even with the 21-plex experiment, we were able to identify the viruses correctly. And since we had different levels of probes in the experiment, it did not lead to the saturation of the probes. So the conclusions so far are Vircap-seq Vertebrate showed up to 1,000-fold increase in reads mapped to 8 candidate viruses. It had higher sensitivity than conventional pre-processed samples, and it showed higher sensitivity and specificity, and with significant reduction in host background. It had lower limit of detection than real-time PCR, and it performed well with higher multiplexing, and probe saturation was not observed. So then we proceeded to validation with real samples using 2 case studies. So one was bat feces sample with rotavirus, and one was deer mouse sample with a novel hepacivirus that was already obtained using race PCR in the lab. So with the bat feces samples, we can see that even without no treatment, capture sequencing did much better than the conventional assay, where we had applied pre-treatment and the DNase treatment. And 9 out of 11 genomic segments of rotaviruses could be recovered, and 2 missing rotavirus segments could be partially recovered with Vircap-seq. And it also recovered partial genomes for other viruses that were not seen in unbiased or the conventional sequencing. And the insect viruses were reduced in Vircap-seq in contrast to conventional. And here is the result for a novel hepacivirus. We can see that we were able to get pretty good coverage. With unbiased sequencing, we were not able to recover this and had very few reads. But here we can see that even with no treatment, we have 49 reads that were mapped, and there is a detection of almost like 1,781 bases. So currently we have moved from NimbleGen to Twist platform, and we modified the probes. And this table just shows the comparison between NimbleGen and Twist platforms. but I'm not going to get into the details of this. But just want to mention that it has been successfully applied to several small and large projects, and the probes have been redesigned using longer probe length using Twist platform, and the CLIA approval for this particular system is underway. And there are several publications out there that support this particular system. And if you're interested, you can check it out. And I would like to end by saying a big thanks to Rafal from CII and the entire team who has been involved in both TBD-CAP-seq and as well as VIRCAP-seq. And I would like to thank you for your time. Thanks.